Hello and welcome to the first longevity review of 2025, where we will take a look at reversing the senescence of senescent cells to increase mouse lifespan, the discovery and beneficial effects of a youthful subset of stem cells which also increase mouse lifespan, the most comprehensive study of life-extending molecules in the roundworm C. elegans, and in the Canadian content study, how disrupting the antioxidant defenses in a specific organ in C. elegans can increase its lifespan. So, a bit of a life extension special this month. My name is Chris Linnell, and I am the founder and president of the Canadian Longevity Association, Canada's premier longevity nonprofit. If you'd like to find out more about what we do, then please check out the links below this video, where you will also find links to the studies being reviewed here. Please also don't forget to like this video and subscribe to our channel. But before we look at the first study, I should briefly mention that this video is a compilation of my new weekly video series called Longevity Snapshot, which you can also find in our channel. So if you would like shorter and more timely updates, then please check them out. So without further ado, let's take a look at the first study. In this study, researchers found a new way to deal with senescent cells. As a reminder, senescent cells are cells which have stopped dividing and have entered a dysfunctional state where they release all sorts of inflammatory molecules causing problems in the rest of the body. This has been a major area of aging research, and traditionally, two methods have been used to deal with them. One method, called senolytics, involves removing those cells. However, that can kill off other properly functioning cells. And the other method, called xenomorphics, involves keeping those cells alive but trying to reduce the inflammatory molecules they release. Here, the researchers have discovered a third method, which involves returning the senescent cells back to a fully functional state. They did this by restarting the cell cycle, whereby cells can once again divide and grow. To show you what I mean, let's take a look at the graphical abstract. So looking down here, you can see the cell cycle. Now, cells first grow in size, then they duplicate their DNA, grow some more, and then divide. And the process continues as the cells proliferate. In senescent cells, there's impaired DNA synthesis, which basically stops this growth cycle, causing the cells to become senescent. And the researchers found a way to restart that cell cycle and thereby rejuvenate the cells. They accomplished this by giving mice exosomes taken from human embryonic stem cells containing high levels of a microRNA called 302B. Exosomes are small spheres of chemical information that cells use to communicate with each other. And microRNAs are molecules which regulate gene expression. Without dwelling on this abstract, let's take a look at the paper itself. And they started off with cells in a dish. Looking at the study design here on the top left, they took fibroblast cells, which are connective tissue cells, and let them divide a number of times. Now, after 30 cell divisions, the cells have yet to become senescent, so those were the controls. However, after 50 cell divisions, the cells do become senescent, and they either gave them a salt solution or these exosomes taken from human embryonic stem cells. And you can see in this image here in B, on the left are the controls, in the middle are the salt solution cells after 50 divisions, and on the right are the exosome given cells. So this is a staining for senescence, and after 30 divisions, you can see there's no blue staining at all. After 50 divisions, the salt given cells have quite a bit of blue in them, indicating high levels of senescence, whereas the exosome given cells have much lower levels of blue, which can also be seen in this bar graph on the right, left being the controls, middle being the salt given cells, and on the right, the exosomes. And there are much lower levels of senescence in the exosome given cells. And in D here, looking at two markers of senescence, P16, P21, and three inflammatory markers, levels were much lower in the exosome given cells. So those are the green bars compared to the control red bars. And looking at G, how well the cells could proliferate, in other words, divide and grow. The top line shows the cells after 30 divisions, and you can see that they continue to grow and proliferate rapidly. The bottom line shows the older control cells, and as you can see, they don't really grow or proliferate that much. Whereas the exosome cells in the middle proliferate considerably more than the controls, though not as well as the younger cells. And in H and I here, we see levels of Ki67, which is a marker of proliferation, and the green bar is much higher than the red bar, once again showing that they could proliferate more, though once again not as high as the younger cells on the left. The bottom here shows differences in gene expression between the exosome cells and the control cells. Next, briefly looking at single cell resolution. And I'll skip over most of this, but I just wanted to show you differences in the cell cycle between the salt-given cells and the exosome-given cells. And as you can see, this light blue section of these bar graphs is higher in the exosome-given cells. And that is part of the cell cycle where the cell copies its DNA. So this is also a marker of proliferative ability. The analysis here shows that the cells could proliferate once again. Now we move on to mice. They used 20-month-old mice, which is roughly the equivalent of a 60 to 70-year-old human, and they gave them weekly injections of either a salt solution or the exosomes, tested them at 30 months, and then let them live out their lifespans. And as you can see in B on the right, the exosome-given mice lived longer. Now, in all of these graphs, green means the exosome-given mice, and red means the salt solution-given mice. And the exosome-given mice lived on average 5 months longer, which is an 18% increase. Looking at their weights in C here over the first 10 months of the study, you can see that whereas the control mice continued to lose weight, the exosome-given mice actually increased their weight. And considering the fact that mice tend to lose weight as they grow older, this is a positive development. Next, they looked at their physical performance. And in D here, the experiment was how long mice could stay on a rotating rod, which increased its rotational speed over the course of 300 seconds. And the exosome-given mice managed to stay on this rod considerably longer. In a second similar experiment, where the rotating rod had a fixed speed of 25 rotations per minute, once again, the exosome-given mice managed to stay on this rod considerably longer than the control mice. Their grip strength was also significantly higher compared to the control mice. Next, to test their cognitive abilities, the mice were placed in a bowl of water, I guess, and they had to try to find a sunken platform to sort of get out of the water. And the mice were placed in quadrant 1, with the sunken platform being in quadrant 3. And the control mice swam quite a bit around to try to find this platform, whereas the exosome-given mice found the platform more quickly, as can also be seen by this graph in H here. So G and H was the training phase, and the exosome-given mice learned to find that platform much more quickly. And in I in J was a memory test. So the mice were once again placed in the water. However, there wasn't a sunken platform. So the mice were tested to see how well they remembered where the sunken platform should have been. And in these heat maps, whereas the control mice swam all over the bowl of water, the exosome-given mice swam mainly in the area where the sunken platform had been, which can also be seen in the bar graph in J here, where again the sunken platform was in quadrant 3, and the exosome mice swam the majority of their time there. Next, looking at inflammatory markers in K. And we have young mice on the left, the control old mice in the middle, and the exosome old mice on the right. And for all 4 of these inflammatory molecules, levels were much lower in the exosome-given mice. In L, we're looking at senescence once again. And as you can see, the young mice have little or no blue staining, showing low levels of senescence. The aged control mice have high levels of blue, indicating lots of senescence, whereas the aged exosome mice have much lower levels of this blue staining. And the same can be seen in these bar graphs to the right in the various organs: kidney, liver, lung, spleen, and skin. And once again, the red bar is much higher than the green bar. The green bar is not as low as the young controls. Next, the researchers looked at individual cells taken from these mice. And we'll just briefly look at this, starting with A in the top left. So cells taken from either the liver or the skin between the old control, the old exosome, and the young mice. And this is the cell cycle. So once again, S is when the cells copy their DNA, and the length of time spent in the S phase was longer in the exosome-given mice comparable to that of the younger mice, both in the liver and in the skin. And looking at F in the bottom right here, once again in the liver and skin, and these are markers of either senescence, so P21 or CCNG2, and markers of proliferation, that Ki67 again. And here the light blue are the old controls, the red are the exosome mice, black are the younger mice. And in each of these cell types, the exosome-given mice were much closer to the young mice than the old controls, as can be seen by the fact the red bars are much closer to the black bars as opposed to the light blue bars for each of these. Next, the researchers wanted to see what was causing this. And again, without going into too much detail, taking a look at A in the top left. So this shows levels of various microRNAs in the cells, and they ranked them according to their abundance, or how much of these microRNAs were in the cells. And the microRNA which had the highest levels was called 302B, and it was considerably higher than any other of the microRNAs. And this microRNA has a role in cellular reprogramming and maintaining the stemness of, stem cells. And when given on its own, this microRNA 302B, and I'll just call it 302B from now on, significantly reduced the levels of senescence in cells that were given it, as can be seen by this lower bar graph on the right compared to this one here on the left. Next, they wanted to see what would happen if they gave exosomes containing high levels of 302B to mice. And this time they didn't take exosomes from human embryonic stem cells. They just took exosomes from regular cells and basically managed to increase the levels of 302B inside them before giving them to the mice. And in this experiment, they started with 25-month-old mice and followed them until they were 30-month-old, at which point they were terminated and analyzed. And I should mention that they use black 6 mice, which are just normal mice, the most common mouse used in experiment. Now let's make this a bit bigger so that you can see. And in C here, you can see the visual differences between the mice. So on the left are the aging control mice from 25 to 30 months, and on the right are the 302B given mice. And you can see that the mice are starting to lose their fur on their backs, which got worse in the control mice. However, for the mice given exosomes from just before they were given the exosomes to 5 months later, you can see that actually their fur regrew. And next, looking at weight, and rather annoyingly, the authors have flipped the colors around. So this time the control aging mice are in green and the exosome given mice are in orange. And you can see that the 302B given mice maintained their weight much better than the controls. The time they managed to hang on to a rotating rod also increased, reaching high levels of statistical significance, as can be seen by the stars above here. The more stars indicating greater statistical significance. And just like this rotor rod test, their grip strength also got better compared to the control mice, whose grip strength declined throughout this period. Moving on to cognitive abilities. Once again, the mice were trained to find a sunken platform in a pool of water. And at the top here are the controls, the bottom are the 302B mice. And at the start of the study, both sets of mice swam all over the place to try to find the platform in the third quadrant. However, 5 months later, whereas the control mice continued to swim around everywhere, the 302B mice swam almost directly to that platform. And you can see in this bar graph, the time taken to find that platform was considerably less as time went on compared to the controls here. On to the memory portion. At the beginning, both controls and treated mice swam around everywhere looking for the platform that was no longer there. However, 5 months later, whereas the control mice still swam around everywhere looking for the platform, the treated mice stayed mainly in the third quadrant. And again, these are sort of heat maps showing the amount of time they spend in a certain area. And this can also be seen in the bar graph, which shows how much time they spent in that third quadrant. And once again, it was considerably higher than the control mice as time went on. Didn't really change much with the control mice. They spent longer amounts of time in that quadrant over the course of the 5 months. Next, they looked at inflammation and senescence. So starting with inflammation and looking at these 4 inflammatory markers in eye, for each of them, levels were much lower in the treated mice than in the controls, and it reached high levels of statistical significance. Looking at senescence, on the left here are stained cross-sections of these various organs, and the blue staining indicates senescence, and in the control mice, there's a lot of blue. However, in the treated mice, there is much less blue, as can also be seen in this bar graph, again with the 5 different organs, and the orange bars are significantly lower than the green bars, again showing that there is a lot less senescence in these organs. Finally, they looked at other markers of senescence and proliferation, once again in brain, liver, and skin. And just a reminder, P21 and CCNG2 are markers of senescence, and Ki67 is a marker of proliferation. And in each of these, the markers of senescence were lower in the treated mice, and the markers of proliferation were higher in all 3 organs. So altogether, these experiments showed that this treatment improved their physical appearance, their performance, their cognitive abilities, as well as lowering levels of inflammation and senescence. However, what about their lifespan, since these mice were euthanized at 30 months of age? Well, that is exactly what these researchers looked at next. So starting with the study design, and I'll make it bigger once again, instead of using 25-month-old mice, they went back to using 20-month-old mice. So once again, that's between the age of 60 and 70 in human years. And every 2 weeks, they gave the mice either a salt solution or these enhanced exosomes containing higher levels of 302B. And they continued giving them the treatment until all the mice had died. One thing I really liked about this experiment was that a relatively large number of mice were used, 80 in each group. Now, quite often in these types of experiments, only a small number of mice are used, which means that it's harder to reach statistical significance. In addition, an equal number of male and female mice were used. Once again, quite often only one sex is used due to cost reasons, and therefore it is impossible to know if there are any differences in lifespan according to sex. Before looking at lifespan, let's see how their body weight changed over the course of the study. So in these graphs, the red represents the exosome 302B mice, the blue represents the controls, and the treated mice maintained their body weight much better than the controls. Moving on to lifespan now, and starting with the combined male and female mice, you can see that the treated mice lived considerably longer than the controls, as we can see by the survival curves. In fact, the median lifespan, or the average lifespan, was 15% greater. Looking at the sex differences, starting with the male mice, the treated mice lived on average 22% longer than the controls. As for the female mice, they lived on average 14.9% more than the controls. Looking at maximum lifespan, in D here, which was when 15% of the mice were still alive. In the combined group, maximum lifespan increased by 12.13%. For the men, it increased by 9.86%, and for the females, it increased by 13.98%. So both for average and maximum lifespan, these are quite impressive results. However, one potential issue with reversing senescence and allowing the cells to proliferate again or divide again is the fear that this might cause cancer, because after all, cancer cells proliferate. And this is true especially in mice, since they tend to die of cancer if they live out their natural lives. What the researchers found, however, was that there was no real difference between the control and the treated mice. There was no significant difference at all, as you can see by these 2 bar graphs here. Likewise, when they came to look at the number of diseases a mouse had when it died, there was no difference between the controls and the treated mice. So although they lived longer, they seemed to die of the same causes. And when looking at the different types of tumors the mice had, there was once again no real difference. I mean, maybe the treated mice didn't have any sarcoma, maybe they had a bit more lymphoma, but overall there was no statistical significant difference between the control and the treated mice. And at the bottom here, this simply shows that in the 4 organs, liver, kidney, lung, and skin, they showed that the treated mice had restored hallmarks of proliferation and reduced hallmarks of inflammation. So what does this all mean? Well, firstly, the researchers have shown that there is now a third means of combating the negative effects of senescent cells, not by getting rid of them, not by reducing their inflammatory markers, but by restoring the function of the cells and restoring them to a more youthful state. It also once again shows the power of exosomes, which I believe is an underappreciated avenue for tackling aging and provides us with a promising avenue for longer and healthier lives. Moving on to the second study. In this study, researchers found that there is a subset of hematopoietic or blood stem cells which retain youthful characteristics even in old age, and when transplanted from old animals into other old animals, can provide numerous benefits, including lengthening their lifespan. With aging, our stem cells become increasingly dysfunctional, and that is one of the main drivers of aging. Previous studies have shown that if you give young blood stem cells to older animals, they live longer. However, with transplantation from other animals, there is the issue of compatibility and the risk of rejection. However, if we could use some of the stem cells already found in our bodies, then we could get around this issue. What the researchers found was that not all stem cells age in the same way, and that there is a subset of those stem cells which retain their youthful characteristics. And if that subset could be reintroduced, then we would get around those compatibility issues. So let's take a look at what the researchers discovered. In the first experiment, they used young mice, and they were given a mixture of blood stem cells taken from young and old mice. Now, this was done twice, and the mice had to be irradiated first to get rid of their existing stem cells. And the first time they did this, it was a mixture of 1 to 2 young to old stem cells, and the second time, it was a mixture of 1-to-1 ratio young to old stem cells. And on the right here, we can see how well they engrafted or survived in their new hosts. And the first time this was done, again, the ratio was 2-to-1 old to young stem cells. And at first, there were more of the old ones compared to the young, but then over the course of 120 days, there was an increasing number of young compared to the old stem cells. And now the second time this was done, when there was a ratio of 1-to-1, initially they were pretty much the same percentage, but then again, over 120 days, the numbers of young compared to old increased, overall indicating that the young stem cells showed better engraftment. Next, they used 13-month-old mice, where they gave them either only young or only old stem cells. Once again, they had to irradiate the mice first, and then after 5 to 8 months, they tested the mice. And what they found was that the young blood stem cells provided a more youthful blood profile. So you can see here, starting with the percentage of B cells. So the young stem cells are in blue, the old ones are in red, and there were higher percentages of B cells. However, there were lower percentages of T cells and myeloid cells. In addition, in E here on the left, there are more white blood cells and lymphoid cells. Taken together, this is a more youthful blood profile. In terms of the immune system in F here, the percentage of naive T cells was greater with the young blood stem cells. T cells attack pathogens, and if they are naive, that means that they have not encountered a pathogen yet. So a greater proportion of naive T cells means that the immune system is more primed to respond to a new threat. On the right, they also found that the blood epigenetic age was reduced in the mice given the young stem cells. Next, they looked at how their physical function was affected. And starting on the left, you can see that their grip strength was greater. The time taken to descend a pole was less, and their ability to cross a small beam was also shorter in the young stem cell given mice. Their ability to hang on to a rotating rod was also greater, and the distance they traveled in the open field test was also greater in the young stem cell mice. On the right, they looked at their memory, in this case using fear conditioning. So in the training phase, the mice learned to associate a tone with an electric shock, or I guess a mild electric shock. And then in the test itself, they were just given the sound and they were tested to see how long they froze, anticipating another electric shock. And here the young stem cell mice froze for a longer period of time than the old stem cell mice. So overall, giving young blood stem cells was beneficial. However, that was already known, so let's take a look at the novel findings from these researchers. Now, it is already known that as stem cells age, they become more diverse. And next, the researchers looked at that diversity. And they did that by looking at the RNA transcriptomal differences between old and young blood stem cells. I won't go into too much detail in this, but if you look at B here, you can see the old ones are in red, the young ones are in green, and they are sort of quite distinct. It's not much overlap between the red and the green here. And as mentioned before, looking at D here, there was much greater diversity in these old blood stem cells, as you can see by sort of these different colors here. And in F here, you can see that according to various markers, there was also a difference between young and old stem cells. And they also found that in the old stem cells, their aging score was higher. They also found that there was a subset of old stem cells which still had that more youthful RNA transcriptomal profile. Next, they looked to see if they could find a marker which differentiated between the younger or older old blood stem cells. And looking at the genetic differences, so they saw that there were 278 genes which were upregulated in the older stem cells, and there were 96 genes correlated to the aging score. There was an overlap of 54. 26 of those were located on the membrane or the outside of the cells. So that could act as a sort of a marker. And they narrowed it down to 7 genes. And out of those 7, they chose one, CD150, for which there were more antibodies, or they could basically analyze it better than the other 6. And what they found was that it was much more prevalent in older mice compared to younger mice. This is the levels of CD150 in the cells. And there was a very high correlation between levels of CD150 and the blood stem cell aging score. As you can see, this line goes pretty much at 45 degrees. They then looked at genes which were associated with aging and whether or not they were up or down regulated. And red means higher regulation, and blue means sort of less so. And from left to right, the amount of CD150 found in the cells. And in the cells which had less CD150, there was greater downregulation of aging genes and less upregulation of those genes. However, in cells which had more CD150, there was less downregulation and much more upregulation. Looking at the epigenetic age, low levels of CD150 resulted in a lower epigenetic age. You can see the blue bar is less than this beige bar. They next looked at the accessibility of genes in our DNA and comparing young to old mice and also low to high CD150 mice. And just looking at these sort of spikes, you can see that the low CD150 was much more closer to the young mice compared to the old mice, and the high CD150 mice were much similar to the older mice in general. And this was the same in both open and closed regions. I guess open meaning more easily accessible, and closed regions meaning it's harder to access the actual genes. Next, they took either low or high CD150 blood stem cells from old mice and gave them to other mice, once again doing this twice. And looking at the engraftment, we can see the blue line being the low, red line being the high, and both in the first and in the second transplantation, there was much higher engraftment of the low CD150 stem cells compared to the high CD150 stem cells, again indicating that they were more youthful. Next, they looked at the differences between these low and high CD150 stem cells. And without going into the details, what they found was that the old CD150 stem cells could self-renew, but they couldn't differentiate as well as the low CD150 stem cells. And differentiate means being able to turn into the various different cell types. So that's, of course, what stem cells do. They have the ability to turn into all the various different types of cells. In this case, the blood stem cells can turn into all the various different types of blood cells. Next comes the very interesting part. And here they took the blood stem cells from old mice, 24-month-old mice, and gave them to other old mice, slightly younger, but still relatively old mice at 13 months of age. And they gave them either all low CD150 blood stem cells, the regular ratio or the whole blood, or just high CD150 expressing blood stem cells. And after 5 to 8 months, their blood and physical performance was tested. And starting with the blood, now in each of these charts, the light blue are the low CD150, purple is the whole blood, and the sort of the beigey red is the high CD150 containing blood stem cells. And in each of these, you can see that there is a gradated difference going from low to whole to high containing cells, and there were more B cells, less T cells, and also less myeloid cells. And with the complete blood count, there are more white blood cells, more lymphoid cells, more neutrophils, more eosinophils, more red blood cells, and also a lower mean corpuscular volume, all indicating a more youthful blood profile with lower levels of CD150. Moving on to the immune cells, and once again, the percentage of naive T cells or naive CD8 plus T cells was much higher in the low CD150 cells. And looking at 2 sets of T cells which have been exposed to pathogens, there were lower levels in the low CD150 cells. Next, looking at physical performance, once again, we can see that in the low CD150 mice, their physical performance was better than in the old CD150 mice. So you can see here on the grip strength, wire hanging, how long they can sort of hang on a wire, once again, how quickly they can cross a beam, or how quickly they can descend a pole, or how long they can stay on a rotating rod, or how long they can basically go on a treadmill before they get exhausted. In all these cases, the old mice given the low CD150 stem cells performed better. And only in the treadmill test did that improvement not reach statistical significance. So yeah, the p-value is over 0.05. In all the others, however, it did reach statistical significance as indicated by either 2 stars or 1 stars, or even 4 stars here for grip strength. Now it gets really exciting, at least for me. And looking at F here, you can see the epigenetic age of the mice, which was much lower in the low CD150 mice compared to either the whole or the high level CD150 mice. And once again, that was a significant difference. And here in G, we can see how that translated into actual lifespan. And in these survival curves, blue are the low CD150, gray are the whole blood transplants, and the beige are the high CD150 transplants. And as you can clearly see, the blue survival curve is further to the right of the other two. Both the median and maximum lifespans were greater. And in percentage terms, there was a 9.4% increase over the whole blood stem cells and an 11.5% increase over the high-level CD150 mice. And in terms of maximum lifespan, there was a 7.2% increase over the whole blood stem cell mice and a 16.9% increase compared to the high CD150 mice, which is not too shabby. In the final set of experiments, once again, 13-month-old mice were given blood stem cell transplants from 24-month-old mice. And this time they were given different ratios of CD150 cells. They were given high or low CD150 cells in different ratios, either 1 to 5, low to high, 1 to 2, low to high, or only low CD150 cells. And starting with blood, once again, and in all these charts, the dark blue are the sort of the control 1 to 5 ratio, light blue are the 1 to 2 ratio, and the sort of light green are the only low CD150 cell transplants. So just to add that the ratio of 1 to 5 is the control because that is the ratio that was found in the older animals. And looking at the results, once again, the percentage of B cells was higher in the pure ratio of low CD150 cells. Compared to the previous experiments, the middle ratio was actually had higher levels of T cells, though the researchers don't sort of mention the significance or lack thereof of that. And the myeloid cells were lower, as we also saw in those previous experiments. Looking at the other components of blood, the white blood cells, neutrophils, and lymphoid cells were higher in the pure low CD150 cells. Looking at the immune system cells, once again, the pure ratio of low CD150 mice had greater proportion of naive T cells than the control. However, the middle ratio had an even higher percentage, and once again, the researchers don't mention in the text the significance of that. However, the memory T cells was lower than the controls, as you can sort of see here, the green bar being lower than the blue, though it did not quite reach statistical significance. Finishing off with their physical performance, and you can see here that the pure low CD150 stem cell given mice performed better than both the whole and the control mice, both in grip strength, the pull test, again, how quickly they can go down a pole, beam balance, thicker beam compared to previous ones, and how long they could stay on a rotor rod. And here's a look at this open field test. It just shows how far the mice travel in this open area, basically. And again, the pure low CD150 given mice traveled a lot further than the controls or the middle ratio. And altogether, what this shows is that the greater proportion of low CD150 blood stem cells given to the older mice, the more youthful their blood profile, as well as having better physical performance. So what does this all mean? Well, firstly, and this is still theoretical, we could take our own blood stem cells out of the body, expand them, separate them according to how much CD150 the cells have, and then put the lower CD150 cells back into our bodies to potentially give us some degree of rejuvenation. Alternatively, if we could simply remove our high CD150 blood stem cells, that might have the same effect, much like removing senescent cells, which show similar benefits. Next, we have the third study. The following study caught my eye because it seems to be the most comprehensive study looking at all the variables going into C. elegans longevity research that I've seen so far, and it throws up some intriguing questions for longevity research in general. In this study, researchers evaluated 16 molecules well documented for extending C. elegans lifespan. They tested them under various conditions, as well as combining some of them to see if there were any synergies, tested some of the best performing ones in the fruit fly to see if there was any cross-species applicability, and also did a screen where they identified some new life-extending molecules in C. elegans. So let's take a look at the first figure. Here you can see the 16 molecules tested from aspirin in the top left all the way down to urolithin A in the bottom right. And in each of these survival curves, the black line is the control and the blue line is with treatment. On the right-hand side, you can see the median lifespan results of those 16 molecules under different concentrations. The first variable which they tested was the food given to C. elegans. C. elegans feeds on bacteria, and the strain of bacteria is called OP50. And in this case, they were given live OP50 bacteria to munch on. Now, you should see that while these 16 molecules were all documented to increase the lifespan of C. elegans, not all of them did so in this test. So, for example, aspirin, the two lines are basically the same with glycine, and perhaps surprisingly, the same with rapamycin. Now, I won't dwell on this first figure because the results are shown again in the next one. The second variable which they tested was, instead of giving C. elegans live bacteria, they gave them dead bacteria to eat. And you can see the survival curves of the same 16 molecules in B, with the median lifespan results under varying concentrations in D. In C, and I'll make it bigger so we can see better, you can see the median lifespan increase between the live and UV-killed bacteria food. For some, such as aspirin, there was no increase in lifespan either for the live or dead bacteria food. Some, however, saw an increase in lifespan when given live bacteria, but that increase disappeared when they were given dead bacteria, such as captopril here, which is an ACE inhibitor. The reverse was also seen, where there was only an increase in lifespan when the animals were fed dead bacteria, as simvastatin shows here. For 6 of them, there was an increase in lifespan under both conditions. These were caffeine, doxycycline, GSK 2126458, which I'll call GSK from now on, LY 294002, which I'll call LY from now on, metformin, and resveratrol. As you should also notice, more of the molecules showed a lifespan increase when the worms were fed live bacteria than dead bacteria. One reason for that could be is because some of the molecules might protect C. elegans against the bacteria, because other studies have shown that C. elegans lives longer when given dead bacteria compared to live bacteria. Not only is there a difference in lifespan depending on the food source, but also in the solvent used to dissolve all of these molecules, which you can see in A on the left here. DMSO is the standard solvent used, and on its own, it can also increase C. elegans lifespan, as you can see comparing the blue line with DMSO compared to the control line here. And also the concentration used has an effect. As you can see on the left, it's 0.1%, and on the right, it's 0.5%, and the 0.5% concentration increases the lifespan more. Now, above 5%, it actually kills the worms. The researchers also looked at what effect these 16 molecules had on the bacteria used for food. And out of the 16 molecules, only resveratrol, LY, and urolithin A had no effect on the bacteria, adding yet another wrinkle to these studies. Next, they took 4 of the best performing molecules and tested them in pairwise combinations, once again using live and dead bacteria as the food source. You can see the lifespan curves at the top here, but the results are clearer when looking at the graphs at the bottom. For a couple of them, there was a synergistic increase. So for example, doxycycline and GSK and LY and GSK showed a considerable increase, as you can see by this blue line compared to the black lines, which are those molecules on their own. Those 2 results are for live bacteria, and for the dead bacteria, only resveratrol and doxycycline showed a synergistic increase. Others didn't show a synergistic increase, and in fact, for 2 of them, resveratrol and GSK and resveratrol and LY, in fact, showed a marked decrease compared to the molecules on their own. And in fact, resveratrol and GSK showed a reduction in lifespan. Next, they tested 2 of the molecules, LY and resveratrol, in 3 other organisms. One, Ju775, which was a different strain of C. elegans, and also C. tropicalis and C. briggsea, once again with live and dead bacteria. So you can see the survival curves on the left-hand side, and here on the right shows the median lifespan. And they had differing results dependent on the organism and the type of bacteria used. So both LY and resveratrol increased the lifespan of Ju775 with live bacteria. However, resveratrol didn't increase it under the dead bacteria. Tropicalis only had its lifespan increase with the dead bacteria. And C. briggsea had an increase with live bacteria. But when they were fed the dead bacteria, they supposedly tried to climb out of the Petri dishes. They didn't like the taste, I guess. That's why there's no graph here on the bottom right. In C here, they also tested LY against 2 mutant worm strains, AGE1 and DAF2, and AGE1 had the gene P13K knocked out, and DAF2 has the gene IGF1 knocked out, and you can see there are long-lived strains of C. elegans, but in the conditions tested, LY only increased the lifespan of AGE1 worms fed live bacteria. You can see here in this survival curve. Next, they looked at giving 7 of the molecules to C. elegans at different stages in its life cycle. According to the authors, and I didn't know this before, most drugs are given to C. elegans starting at L4, which I believe is the 4th larval stage. And in this experiment, they gave C. elegans the molecules 1 day after they became adults, once again with live and dead bacteria, as you can see in the survival curves at the top. Looking at the median lifespan between L4 and day 1, for the live bacteria, doxycycline showed a comparable increase in lifespan between L4 and day 1, whereas the others showed a decrease. For the dead bacteria, once again, doxycycline showed a comparable increase, as did caffeine and GSK. The other 3, once again, showed a decrease. Next, they tested 5 of the best performing molecules also in fruit flies. And nice to see that they also used both male and female fruit flies to see if there was any sex differences. And you can see the survival curves at the top. The median lifespan increase under varying concentrations in B, And we'll take a close look at the results in C here. So as you can see, doxycycline showed a comparable increase in lifespan across species, across the sexes of the fruit flies, and across the two different food sources. Metformin also showed a comparable improvement. However, LY didn't show any increase in female flies. Monocycline only showed an increase in live-fed C. elegans. And whereas rifampicin only showed an increase in live-fed C. elegans, it also showed an increase in male and female fly lifespan. Lastly, they did a screen of hundreds of molecules to see if they could find any life-extending ones among them. And you can see the results here at the top with the live bacteria-fed ones and the UV-killed ones on the right. And the results showed either increases or decreases in lifespan. Now, out of the live bacteria-fed C. elegans, only 34 out of the 470 molecules tested showed a lifespan increase. And in the dead bacteria-fed C. elegans, only 39 out of the 592 molecules tested showed an increase in lifespan. And that was either 7.2% or 6.7% out of all molecules tested. Then they tried to replicate those positive results numerous times and came up with 11 molecules showing robust extension for the live-fed C. elegans and 6 molecules for the dead bacteria-fed C. elegans. And out of those, 5 molecules increased lifespan under both conditions. Looking more closely at C at the bottom here, those 5 molecules were the antifungal mecanizole with a 26% increase in the live-fed and a 23% increase in the dead-fed. The antiparasitic fenbendazole with an 8% increase and 57% increase. The PDE5 inhibitor tadalafil, better known as Cialis, showed a 28 and 26% increase. The MAO-B inhibitor used in Parkinson's, safinamide, showed a 36 and 14% increase. And lastly, the tyrosine kinase inhibitor, AG1478, showed a 14 and 17% increase. Now, out of those 5 molecules, only AG1478 had been characterized as being a lifespan increasing molecule. So the other 4 are new molecules in that regard. So what does this all mean, and why did I think this had profound implications? Well, firstly, it shows that you can't trust single studies in C. elegans on their own. This study showed that there are so many variables which can influence C. elegans' lifespan. The concentration of the solvent, whether or not their bacterial food source is dead or alive, the influence the molecules have on the bacteria themselves, at what stage in C. elegans' life cycle the molecules are administered, the concentration of the molecules, not to mention that different species of roundworms have different results, and that the results might not cross the species barrier. It has certainly made me much more wary of trusting any C. elegans lifespan results. And I hope that Matt Kaberlein and his million molecule challenge, where they're using the worm bot to try to screen as many molecules as possible in C. elegans, has taken note of this paper. I'm sure he has. Finally, and this pertains to us, be very careful when trying to combine supplements. As we've seen, molecules which on their own increase lifespan don't necessarily do so when combined. In fact, most of the combinations did not lead to an increase in lifespan. Indeed, some of them actually led to a decrease. The ones which synergized effectively were few and far between. So please be careful out there. And finishing off with the Canadian content study. Now, before we go into the study, a little bit of background on free radicals. So free radicals, also known as reactive oxygen species, or ROS, are produced by our body's metabolism. Now, our bodies have a defense against these free radicals, such as the antioxidants glutathione and superoxide dismutase. The free radical theory of aging, one of the oldest aging theories out there, suggests that the damage caused by free radicals is the primary driver of aging. And if we could reduce levels of these free radicals, then lifespans could be lengthened. There are numerous studies which support the theory. However, there are also a number of studies which contradict it. Indeed, in 2009, the authors of the present study found that knocking out one of those antioxidants, superoxide dismutase, or SOD, could increase the lifespan of worms. There is a link to that study as well as a general overview of what I've just said below this video. Today's study is very much a follow-on to that 2009 paper. Here they're looking at what effects turning on or turning off SOD in specific tissue has on the lifespan of worms. So let's look at the paper. The study comes from the lab of Jeremy van Ramsdonk from the University of McGill in Montreal, and we'll start by looking at the graphical abstract. The study used C. elegans, a tiny roundworm which only lives for a number of days. You can see a cross-section of the worm here. And by turning off mitochondrial SOD in specific tissue, that led to a rise in mitochondrial ROS, or free radicals. When this was done in the intestine, it led to an increase in lifespan, an increase in heat stress resistance, however, a decrease in oxidative stress resistance. And you can see the survival curve here, with the black line as control, and the red line are the worms which had the SOD knocked out in their intestine. As you can see, the curve has shifted to the right. Now let's take a look at the study in more detail. First, they looked at what would happen if SOD was only turned on in a specific tissue. And for that, they created a series of worms which only had SOD turned on in the various tissues. Now, the worms also had a fluorescent marker when SOD was turned on. And you can see those worms at the top here. So on the left are the ubiquitous SOD worms, which means that SOD was turned on throughout the body. As you can see, it's sort of lit up here. Then there were germline worms. Those are the reproductive parts of the worm. You can see lit up here. The muscles on the outside of the worms, the intestine on the inside, and also the neurons, so the nervous system of the worms. Looking at B here, which shows levels of SOD, on the left-hand side, the WT stands for wild type, so the normal worms, and they were the index or reference values. Now, the strain of worm they used in these studies is called CLK because it showed a greater lifespan effect when SOD was turned off in that 2009 study. And you can see that its levels of SOD were almost as high as the wild type worms. SOD2 worms are worms which had SOD turned off completely, and as you can see, there's no levels of it whatsoever. And in this study, all the worms were a cross between CLK and SOD2, and you can see that those worms also expressed no SOD here at all. Then come the 5 worms we looked at above, and you can see that their levels of SOD were much lower than the wild type. However, they at least had levels of SOD, unlike the CLK1 SOD2 worms. C simply shows how much of the fluorescent marker was expressed in the various worms. And obviously only the worms which were given the marker expressed it. Next, looking at fertility, and the CLK1 SOD2 cross worms show very low levels of fertility when compared to the wild type worms on the left. And the same went for most of the tissue-specific worms, apart from, unsurprisingly, the reproductive or germline worms, which showed higher levels of fertility. However, when it was expressed throughout the worm's body, fertility increased a lot. When looking at the survival of the baby worms, or lethality, as I should say, in wild-type worms, hardly any died. However, the strain used here had much higher lethality, as you can see by this purple bar, which increased even more when SOD was only turned on in the muscle or intestine. However, it was reduced, unsurprisingly, when it was turned on in the germline. You can see by this small red bar here. The post-embryonic development can be seen in F here, with the black line representing the wild-type worms, and all the other strains developed much more slowly. And you can see here the strain used and all the tissue-specific worms are clustered around here on the right. Only the worms ubiquitously expressing SOD developed faster, as can be seen by this blue line here. And looking at movement or body bends, compared to the wild-type worms in white, all the other strains moved a lot less, with the intestine SOD worms moving the least of all. Next, they looked at how the worms would respond to 3 different stressors. First, they were tested against a pathogen, and the worms with no SOD survived much longer than the normal worms on the left. When the SOD was turned on throughout the body, their survival decreased to levels close to the normal worms. In the worms where SOD was only expressed in the specific tissue, their survival increased again, but it didn't reach the levels where there was no SOD at all. The next stressor was heat stress, where they were exposed to 37 degrees centigrade for 3 hours, and the worms lacking SOD survived a lot longer than the wild-type worms. And the same went if the SOD was only expressed in the germline or intestine. In fact, all 4 of the specific tissue worms lived longer than the wild-type worms. Next, they were exposed to oxidative stress in the form of paraquat, which is a very toxic herbicide. And here, the specific tissue SOD worms lived longer than the wild-type worms, with the neuronal SOD worms living the longest. Next, they looked at lifespan, and starting with A on the top left, the wild-type normal worms are the dashed black line. Worms lacking SOD are the gray line. The CLK worms are the black line, and the CLK worms, which also lack SOD, is the purple line. And as you can see, they live considerably longer. Now, comparing the CLK worms lacking SOD, when SOD was turned on back again, it shortened their lifespans in B here. When looking at the specific tissue SOD worms, expressing it only in the germline saw a small increase in lifespan. The same can be said when it was only expressed in the muscle, intestine, and to the greatest extent in the neurons, which is also shown in the bar graph at the bottom here, which is showing mean lifespan or average lifespan of the worms. They then reversed this experiment. Instead of only turning superoxide dismutase, or SOD, in one tissue, they looked at what would happen if they turned it off in only one tissue. And they used RNA interference for this purpose. Now, RNA, among other things, carries the genetic information from our DNA to where proteins are produced to produce those proteins. And by interfering with that process in specific tissue, you can turn SOD off. So in all these survival curves, the black line is the control, and EV stands for empty vectors. So the worms were given the same treatment. However, in the controls, they didn't have that RNA interference. And in A on the top left here, when they gave CLK1 worms this RNA interference throughout their bodies to turn off SOD, they lived longer, as can be seen by the red line being further to the right of the black. When the same treatment was given to wild-type or normal worms, there wasn't any difference, which shows how the CLK1 worms are more sensitive to SOD. On the right in C are worms which have this RNA interference mechanism switched off, so when they were given RNA interference, it made no difference to their lifespan. Moving on to the worms where SOD was turned off in specific tissue, and starting at H here, which is the hypodermis or skin of the worms, and here turning SOD off in the skin seemed to shorten their lifespan, even more so when it was turned off in the muscle here in G. For the neurons and germline and ENF here, there was no statistical difference in their survival. However, when SOD was turned off in the intestine, there was a marked increase in survival. You can see this is comparable to when it was turned off in the worm as a whole, indicating that SOD only needs to be turned off in the intestine to increase the lifespan of these worms. And D here is the same figure we saw in the abstract. Next, they zeroed in on these worms where the SOD was knocked out in the intestine and looked at how they would respond to the same 3 stressors we saw before. So in all 3 of these graphs, wild type is on the left, the strain used in the study, CLK, is here. The strain with the RNA interference ability, second from the right, and where the SOD was knocked out in the intestine on the right-hand side. And when confronted with a pathogen, none of the 4 strains showed any major difference. In fact, it was all not significant. When exposed to heat stress, the CLK and intestine knocked out worm saw a significant and comparable increase in survival. The opposite occurred when the worms were exposed to paraquat, the oxidative stressor. And here there was a comparable decrease in survival between the CLK worms and the intestine SOD knockout worms. Finally, they looked at the function of the worms, starting with the embryonic lethality in A here. And whilst knocking out SOD throughout CLK worms led to a huge amount of lethality, when it was only knocked out in the intestine, it led to a non-significant amount, though still higher than wild-type worms. When looking at fertility, or the brood size, knocking out SOD throughout the CLK worms led to, it looks like no worms surviving whatsoever, whereas knocking out SOD only in the intestines didn't seem to have the same effect. However, again, this was much less than in the wild-type worms. Looking at the development time of the worms, with wild type on the left here, and knocking out SOD in them made no difference whatsoever. When knocked out in CLK worms, it really lengthened their developmental time. However, the difference became non-significant when SOD was only knocked out in the intestine, as you can see down here on the left. Finishing off with movement or thrashing rate, knocking out SOD in the entire worm or only in the intestines made no significant difference. So returning to the graphical abstract, what does this all mean? Well, the study is quite interesting in that turning off superoxide dismutase in a single organ, the intestine, can result in not only an increased lifespan, but also greater heat stress resistance, though it does also lead to reduced oxidative stress resistance, as well as reduced fertility. Now, in terms of the generalizability of these results, you need to remember that this was only done in a single strain of worm, and that further studies are needed to see if this also applies to higher order animals such as mammals or even humans. This is very basic but necessary biology, so who knows where it ends up leading. As for next steps, the researchers here are working on how elevated levels of mitochondrial SOD affect nuclear gene expression, and they are just completing a paper where they've identified a signaling pathway required for SOD longevity that may act to mediate communication between the mitochondria and the nucleus. At the CLA, we wish Jeremy and his crew the best of luck and hope they continue to make major longevity discoveries. I wish there were more work like this in Canada, and the CLA will do everything in our power to encourage it. And that marks the end of another longevity review. Please don't forget to like this video and subscribe to our channel. I'd also love to hear your thoughts about these studies, so please put them in the comments below. So until next month, goodbye.